OChem and I want to take a nap. Nope, I did not just take an OChem, I took an OChem final.
Okay, welcome back to another episode of this podcast that doesn't have a name and has no other episodes. I'm your regular host, Eva, sophomore chem major at MUD, and I want to introduce my lovely guest, Florence.
My name's Florence. I'm a sophomore at the University of Washington. I'm a bioengineering major, and I just took an OChem, and I want to take a nap. Nope, I did not just take an OChem, I took an OChem final.
That's where we're at mentally, folks. Pretty standard around here. All right, so our topic for today is this lovely biomineralization paper. I say lovely, I wrote it, that feels like tooting my own horn, but the graphics are pretty. The graphics that I took from other papers and cited are pretty cool looking.
I can't see it, but I'll take your word for it.
Gotcha. So one of the things that I want to talk about with this paper is like why we care about corals at all. Because I feel like in grade school we talked about corals and it was just like the Great Barrier Reef, we have to save it, you know?
Yeah.
You remember this.
All the colors are going away.
Exactly. And like, I love the Great Barrier Reef. I think reefs are nice. But also as like a 10-year-old from Utah, I didn't really know what a reef was.
Yeah.
Yeah, and I think you had a similar.
Same.
It was just like, there are colors in the ocean somewhere. And for me, I had been to the ocean once in my entire life. So I was like, this is important? Why?
Why?
I thought the ocean was cold. That was like all I knew about it. And I liked sharks too.
And dolphins.
Dolphins. I love dolphins. Yeah. But anyways, the reason we're talking about corals is because they're really useful as paleoproxies, which is a word
That's a word that's a word.
It's a word. Microsoft Word does not agree with me that it's a word, but it's a word.
Okay.
So a paleoproxy is basically something that we can use to reconstruct part of Earth's climate. So I feel like the standard example for this is tree rings. So like you can look at a tree ring and get insight into rainfall or if the tree got lit on fire or other
so
things.
like by reconstruct you mean like paint a picture, not like physically reconstruct it.
Yeah, well, it would be cool if we could build a little back. Yeah, I feel ya. But on some level, we can kind of physically reconstruct because we can figure out like mineral compositions and things like that. So it's pretty cool. So we can do that with tree rings. We can also do that with like ice cores. If you've seen how they take the gigantic ice cores out of Antarctica and we can look like hundreds of thousands of years back.
Yes.
And I think this is very cool.
That.
Yeah. And the reason that and then the reason this abstract stuff matters is because climate change.
Big oof, yes.
Big oof. That's honestly how I feel like every single presentation about climate leaves me feeling. It's just like, well, this is where we are. Yeah. So the cool thing about corals is that, so if you, and I have to kind of explain like how corals are structured in order to explain why they're good paleoproxies. Because corals build their own skeletons, mostly out of calcium carbonate. We build our own skeletons. Most things the skeletons build them. But the reason that it's really cool with corals is because corals build theirs from the surrounding seawater. And they build them in layers, kind of like a tree. So like if you were to slice open a coral, and I don't recommend doing this if you don't have like a permit or something.
Catch me next weekend.
But if you slice open a coral, you can actually see layers of it. And it'll be like dark, light, dark, light, dark, light. And they have a yearly cycle, which is really cool to see. I can actually send you a picture of it if you want to look at it. Or our lovely listeners can just Google it.
Just Google it.
But it is really cool because you can look back and be like, oh, look, 2005. Why is the coral so messed up in 2005? And then maybe you go back and you look at like your records of temperature or hurricanes or whatever, and you're like, they had a bit of a bender.
Was 2005 actually a bender, or is that just random?
That's just off the top of my head.
Okay, because I was like, I don't remember what happened.
You were 4. How would you remember what happened?
You never know.
Yeah. Okay, gotcha. So corals take the surrounding seawater and they turn it into their little skeletons, which is fun. And the cool thing is that they actually modify the composition of the seawater. And this is where we don't totally understand how it works. But what scientists have done is they've grown crystals just out of straight seawater. It's called aragonite. It makes like this almost salmon-colored crystal.
I've heard that one. I've seen that one.
It's pretty. I'm not gonna lie.
It is pretty.
Literally seawater you left to dry, but it looks really cool. And
not just salt though.
No, there's that's what we're getting into. There's other stuff in there too. So what's actually forming the crystals is not the salt. It is calcium carbonate.
Yes.
Yeah. So most of aragonite is calcium carbonate, but what happens is that other trace minerals get mixed in, like strontium and magnesium are kind of the two main ones. And the cool thing about corals is that compared to seawater, they lower the ratio of strontium to calcium and they raise the ratio of magnesium to calcium.
So magnesium good.
For corals, presumably. And
they're good for humans.
Yep. Although we need strontium too, from what I know, but I don't think we need nearly as much.
We need a lot.
Yeah.
We need a lot. But magnesium stabilizes organic molecules.
Yeah.
Okay,
lovely Chem final over here.
Sorry.
No, it's great. Thank you. So we know that corals can like mess with the ocean water composition already.
Yes.
We don't know how. We're still working on that.
So they mess with the actual ocean water, or just the stuff they're taking? So like they're taking out what they need and that's affecting the concentration, or they're just like choosing what they take in that way? So
that's like how corals make their skeletons. It's really weird the way they make their skeletons. They produce this, it's called a calcifying fluid. My mental image of it has always been kind of like saliva. And then that hardens into their outermost layer of the coral. So that calcifying fluid is, they intake ocean water and then they like kind of spew out this calcifying fluid. And again, that's a process that's not fully understood, and we're going to get into that a little bit, depending on how long we decide to make our podcast. We have very random episode lengths. You never know what you're going to get. It's one of the many trademarks of Untitled Podcast. No other episode.
Untitled Podcast, never heard before, but gonna keep happening.
Yeah. But we know that coral like systematically does this. It modifies stuff. And the cool thing about that is the ways in which coral changes its ratios depend on the temperature of the water that it's in. So that is how we can kind of use coral to reconstruct temperature records. And the really wild thing, at least to me, I don't know, I always think of corals in my head as like being very small and like not super old. We can use corals to reconstruct ocean temperatures for going back a couple hundred years.
Oh, wow.
Yeah. So pretty much the whole industrial era. Which is...
That's useful.
It's honestly really convenient. And the best part with corals is that it's really localized, right? Like with ice cores, it's not telling you like what Antarctic weather was like on Saturday, June 15th, 1923. Like, it's not going to tell you that. Yeah. It will tell you there were more volcanic eruptions this year than usual. Or the ratio of oxygen in the atmosphere dipped slightly, you know. But with corals, it's cool. We can actually see like these strong seasonal fluctuations, and they look similar to temperature graphs.
Are they similar to like CO2 emission graphs? Because those kind of follow along the same trends.
So with CO2 emission graphs, you will see a yearly cycle, but you also see like the terrifying uptick that haunts my nightmares. We don't see the uptick in the same way because they're biological systems, they're self-regulating. So that helps a lot.
But is it the same like oscillation?
It's the same yearly oscillation. Yeah, that's a good way to think about it. So what we do is we take these corals and we take big cross sections out of them. We like pull out slices of ancient corals. It sounds wild to me, especially because, I don't know, in terms of what they're made of, it's almost like teeth, like a giant
tooth.
Yeah, it's like a giant tooth that you're pulling out, and then you split up the sections, and then you can break down the sections using acid and then analyze their chemical composition. So we can be like, there's a lot of strontium in this year. What was going on there? And we've been able to kind of use that to figure out what does that tell us about the way the ocean was during that year. So there's a really distinct marker for when ocean temperatures like jump up. And that causes reef bleaching, which is, again, the stuff they scared us about in, like, 3rd grade. And it was like, the reefs are being bleached. And I don't know how you pictured that. I just literally imagined, like, white coral reefs and their skeletons.
I did, too.
Okay. Yeah.
I did, too.
Yeah, bleaching is...
I still do.
It's not far off. Basically, it's, like, a good term. So when they talk about bleaching, it's a good term for, like, mass damage. Of a reef. A good thing to know, though, is that reefs do actually recover. I thought they didn't recover. I thought, like,
I saw that, like, a year or two ago.
Yeah, they didn't tell us that the reefs could recover. I thought they were just dead.
They were just like, it's all doomed.
Yeah, I had this almost plague-esque concept of the reaches across the world slowly being, like, turned to little kitchen sponges, you know? Didn't make too much sense, but I was not a very bright 10-year-old, so, you know.
Well, I think you were bright.
Thank you. I was annoying. She can attest. She's known me that long, for the record.
I have.
For our lovely audience.
for our monthly listeners.
Yeah. So, anyways, where were we? Before we got kind of derailed. Okay, but yeah, with like bleaching of coral reefs. So they can recover, but these bleaching instances are really useful for us to be able to know about because that happens when temperatures are really high for the region. And so there are regions of Australia, like, this is one of my favorite things is how the Australian regions are named. One region is called the Kimberley region, and it has like incredibly violent ocean patterns and really strong temperature fluctuations. And it's called the Kimberley region. And
I have some issues with that.
So yeah, that's naming it's fun. But Australia is a really good spot to like study this sort of thing. And
I have a question.
Yo, go for it.
So when you're talking about you being like looking at coral and being able to see temperature fluctuations, is it because like from my understanding, but this is also just my understanding, I don't know the actual details. Like it's temperature, but the thing that's actually harming it is the increased CO2 because it acidifies it. and like breaks it down, but it's just like correlated with high temperatures because higher temperatures is higher CO2.
It's both.
Okay.
So, ocean acidification is an issue because it makes it harder for calcium carbonate to stay as its like skeletal structure. Yeah. And also higher temperatures can lead to this bleaching that we see, where it's and we're going to talk about why. We don't fully understand why high temperatures are bad for corals. There are a couple different reasons. One of them that I think is really interesting is remembering that corals are actually in a symbiosis with algae. And so some people hypothesize that it's actually the algae who can't handle the high temperatures. Oh. But again, it's really hard to dissociate like the two halves of a symbiotic relationship when they're always paired. So
together, definitely.
Yeah. So both of them are bad. From what I know, the calcium carbonate is like a more direct threat.
Yeah, I think I'm just familiar with like the acidification. Mm-hmm. So like that's the stuff I've heard. Yeah, at least once before.
Yes. Yeah, the acidification especially scares me because I think people aren't as concerned about it as they should be.
Yeah, it's in all of like the bio tests I've taken in the past 4 years.
I feel ya. Me as well. Actually, that's not true. I had one exam that was just like cholera in the 1800s.
Oh.
That was weird.
That's some lighthearted fun.
Oh, joy. Fun times. Okay. But yeah, kind of get a little bit back on track. So the cool thing is we can see, and again, this is all backwards because it's southern hemisphere, backwards from what we're used to. So like in the summer, we can see, oh, look, it's pulling in more magnesium relative to calcium, and then that ratio will fall a little bit as it gets cooler, just in terms of the water temperatures. We can see those like year to year, and they're really consistent, which is super helpful if you are trying to track like, hey, we don't have good temperature records for this summer because our equipment got wiped out. Or it was 1880 and no one was taking temperature records in Australia. Because I believe at that point it would have been indigenous peoples and then British penal colonies. And Florence, will you tell our audience where you're from?
Hi, my mom is from the UK. To put it lightly.
All right, so it's really, really helpful if we want to go back and reconstruct. But part of the problem is we don't totally understand like why. We've been able to see that these things, like we've got this jump in strontium and a drop in magnesium. We're like, that means there was bleaching and it was really warm. But we don't totally know why.
Why it fluctuates and what it takes in.
Yeah, because we don't understand like the basic mechanism. So it would be like trying to explain anemia if you didn't know that the body needed iron to begin with.
Yeah.
I mean, we're a little farther along than that. There are at least theories, but... So there's a couple things. One is that it's not a matter of like calcification, which is... calcification is just straight up the amount of additional skeleton that it builds up. That doesn't depend on temperature. Or if it does depend on temperature, we can't disconnect it from other things as easily as we can these ratios. There's definitely a yearly cycle. It's how like, it's this very nice light dark banding thing, but we can't just be like, oh, it added 0.2 centimeters. That must mean it was a cold year. We can't do that yet. Yeah. And so we're looking at like why all these different things could be due to part of it. One theory is that there is a really important protein called the calcium ATPase, which I always say is ATPase, but it's ATPase.
Do you remember my HL bio was on ATPase?
Yes, this is part of why I wanted you around. Florence knows ATPase. But that one is also involved in, like, actually pumping calcium ions into the calcium fluid.
Yeah.
So some people theorize that the protein doesn't work as well at high temperatures. And that would explain why you've got this jump in the strontium ratio, because there's not as much calcium being pumped in.
So it's just the ratio, not the actual finite amount.
It's the ratio.
Okay.
Yeah. The amount is due to a whole bunch of things that we can't really figure out yet. So some people are like, yes, if there was just less calcium being added to the calcium fluid, that would explain why the strontium to calcium ratio jumps up. Mm-hmm. But it doesn't explain why the magnesium to calcium ratio lowers, is the issue.
So there's less magnesium?
There's less magnesium relative to calcium than there would normally be.
But there's also less calcium, right? Is that
not
the theory?
Not in terms of quantity, but in terms of, like, relative
ratio.
amounts. Yeah.
So, like, less than strontium, but less magnesium than strontium.
Yeah, it's weird. So a drop explains half of it, but not the whole thing. Some other people think that basically when there's like this sudden warming event, it actually becomes more similar to just straight up ocean water. Because corals normally lower strontium and raise magnesium. So the opposite actually brings you back a little bit closer to regular ocean water.
So, like, when it's hotter, the coral doesn't regulate as much, or?
Yeah, that's okay. Part of the theory. Which, again, doesn't completely explain why. Because, again, we don't know how corals, exactly the mechanisms behind corals controlling these ratios. Because they do it. Quite intentionally, which is interesting, especially because they almost seem like defects in the structure, but it's like a very systematic change. And then the last, kind of the last explanation that I know about, is actually due to the algae, which are one of the coral's like bigger sources of ATP. Yeah. And so if they're just not producing as much ATP for the calcium ATPase, that could explain why it's not denatured, but it doesn't work as well at warm temperatures.
Yeah, so, like, it's not denatured, so it's because it's not, like, boiling hot, but, like, wouldn't high temperatures to an extent increase the rate just because of, like, energetics and kinetics?
Yeah, I think about it in terms of, like, these are already pretty warm waters we're looking at. This is mostly northern Australia. What I'm used to. Yeah, not the freezing Welsh coast. From my understanding, because, like, again, regions like the Kimberley area are a little bit less vulnerable, and corals vary a lot. Like, if you go to a different part, you're going to get something completely different in terms of the types of corals that grow there, their makeup, their composition, but they all have, like, the same rough mechanisms going on here.
Okay.
So, again, it's really frustrating because it's basically like we don't totally know how it works, which is infuriating when I feel like corals are something that, for me, it's really satisfying to be like, you're a climate skeptic? Look at these corals. How could we possibly have faked these corals? How did
we possibly have baked them?
Faked.
Oh.
I mean, they're getting baked at this rate.
I know. I was like, oh.
Dark. There are only so many jokes about climate change you can make before it just gets kind of depressing. Anyways. But yeah, that's kind of where we are with our understanding of corals. We're, like, getting closer. We're getting to a better understanding. But we aren't at the point where we can be like, where we can just say, oh, it's because this protein gets slightly less efficient in warmer temperatures and that sets off XYZ and this whole chain reaction. So we're working on it, but we're still not quite there yet.
So is this like, when you say it's an ATPase, is it like, that's just synthesizing ATP? So what does that have to do with calcium?
Got it. So
Or do you mean ATPase is in like something else?
That is how it was referred to in the literature. They didn't explain if it was responsible for, I think it
provides the energy for it.
I
Because that
think
would then like relate to like the algae thing.
My understanding is that this protein is responsible for providing the energy to pump calcium ions in.
Okay, yeah.
Yeah, but I don't believe it is responsible for exclusively responsible for producing that energy on its own, from my understanding. So it is a little stressful, but I think it's also pretty cool because it's like
cool.
Yeah.
It's so like there's like a yearly like you talk about the layers, like there's like consistent dark and light. What's the dark from?
So the darkening is actually like the density of it changing.
Oh.
So if you think about, and what they'll do is they'll photograph this on like, in terms of like, black and white. So what you're actually looking at is basically the density and like the saturation, and that produces this like dark light, dark light, dark light. Yeah, and that the thickness of the bands can really vary from year to year. That's my understanding. But it is generally like you have a thicker band in summer, and then a thinner band in winter. So it's, yeah, and that's not totally temperature dependent because you've also got like crazy ocean current stuff going on.
Yeah,
and
I was like
thermohalides.
thinking about all the currents. I was like,
Yeah, and I would personally love to look into that more. I don't know a ton about it because you've got some corals that are like really protected and so are dealing with relatively constant waters versus like the absolute wackos in Kimberley who are...
But they're all relatively similar, even with like differences.
Yeah, it's cool. Some of the corals they look at are actually like in the Mediterranean, and you can use the same principles on those as like Australian corals. And,
wow.
I mean, I thought there were only corals in Australia when I was very dumb. I had indeed learned there were corals in other parts of the world. Sometime in, I think, middle school. I was really confident it was just the Great Barrier Reef. It's not, if you're in my boat. But yeah, so we can kind of use this across the world, and it is really cool to be able to see the same things where it's like, oh look, corals in the Mediterranean show the same pattern as they do in the Caribbean and off the coast of Australia and in South America, which is kind of wild to me. So it's really cool to look at.
Yeah. Are there like, can you further the applications? Is this something that you're looking into and you think it can be applied, or is it just something that you're looking into because it is being applied in a cool way?
So I was interested in it because I knew about corals as a paleoproxy. I didn't know like how that worked because I feel like in like I've taken like 1.5 climate classes so far. I'm working on one right now. And a lot of the time it'll be like, we're going to talk about a bunch of different paleoproxies at once. And so we talk about trees, and that's pretty quick. When we talk about lake sediments, and corals are kind of thrown in there, and you'll see like some very cool pictures, but I had never gone in depth as to how that worked. So I just kind of wanted to learn how it worked.
It's definitely cool because like, I mean, I guess trees are an organism too. But I feel like they feel a bit more like landscapey than like a coral does almost, to me at least. So it's cool to see how that can...
Yeah, and corals are especially important because the ocean is one of those things where, so it keeps absorbing CO2 for us, right? We don't know if there's a limit on what the ocean can take, which is terrifying, right? This is one of the things in climate where it's like, how long can we keep pushing this button before we get attacked by the bull? Like, we have no idea.
I mean, I would assume there's a limit, and I assume we're getting there. Just put it in depressing terms.
But yeah, it is it is kind of wild because we can, and so I've like learned a lot about how we can use like isotopic ratios on ice cores, which is one of the big ones that I really enjoy. And like, I was just basically curious if it was something similar with corals. I also really like corals. I would love to work on this sort of stuff someday. Because, like, you get to do science and you get to go scuba diving. Are you freaking joking? It's the coolest job ever.
I get to sit on a boat and I get to be in the sun.
And you have to deal with sharks to get your data.
Yeah.
Sharks are cool.
I'll be dealing with sharks, but, like, the human kind.
We are salty here, but yeah. All right. Well, I think that's all we've got to cover. If you're doing all good, Florence, I know you need a nap after your many exams. Florence graciously...
I still have homework.
I know. Florence was graciously my guest after taking a couple of finals in the last, 24 hours.
Woo!
Woohoo! So, thank you very much for joining me on this.
I had a fun time. It was informative. I learned some stuff. I'm glad I participated. Never thought I'd be a guest on a podcast.
She is not being paid to make these comments. I would like to clarify.
I wish. Trust me, I'd ask.
I can send you some goldfish in the mail.
Oh, I'd be down with that.
All right. So I believe that's the end of our episode. I wish we had a cool podcast outro jingle. Yeah, they always have good jingles.
I can sing one.
Let's hear it. I would love that.
No, I don't. My mind went blank.
Okay.
All the songs I'm normally singing.
Okay. All blank. Ready to go. Anyways, thank you very much for joining us. It's been a lovely time. Thank you for joining me, Florence. This was lovely. Thank you very much.